Okay, hello everyone. This is going to be the recording for lecture 3. Right now it is 8:30 p.m. on the evening of August 31st. Let me go ahead and share screen. Farther up. Okay. So I have a fair amount to get through today. So I'm going to be moving fairly quickly. Just some announcements before we get into the material. Lab 1 and Quiz 1 are due today. Again, I'm recording this as of August 31st. So by the time you see this recording, those should be submitted. As I mentioned this a couple of times in email already, but going forward, live lectures will likely be Mondays and Wednesdays at 8:30 p.m. Not always. I mean, there's going to be times where I might not be able to make it for one thing or the other. It's the online flexible modality. So normally I'm going to be here on this recurring Zoom link, which you can see under your under the lecture module on Canvas. So that's the one I'm on right now. You can feel absolutely free to log in and participate live. I encourage it. Keep me from getting lonely here. But mostly, these are going to take place on Mondays and Wednesdays at 8:30 p.m. With the possibility of some flexibility in there. Let me see. I just want to make sure I'm recording. Okay. Exam availability. As you know, we have 3 midterm exams scheduled in the class, and I know this is a little bit far away. We're only at week 2, but I just want to put it on your radar. Coming down from the dean of the natural sciences, there needs to be a window. I was initially going to put it at a 24-hour availability, but there needs to be a window of availability for exams. Oh, 1 second. Okay, 1 second. Admit. Looks like I get some company. Hello, Morgana and Amanda. Hello. Nice to see you. Feel free. Glad you're here. The other folks may join in as the day goes on. I'll go ahead and start over here. But yeah, glad you're on the recording. Feel free at any point to interrupt me and ask questions. Once I go into screen share mode, just so you know, I can't see the chat menu. Feel free to just ask questions vocally at any point. Let me go back to share screen here. And okay, so just going to start over. This is the evening of August 31st on Monday. So for folks who are watching the recording, Lab 1 and Quiz 1 are due today, I think by midnight, 11:59 p.m. So by the time the recording's up, those will be due. Going forward, live lecture sessions, like what we're doing right now, are most likely going to be Mondays and Wednesdays at around 8:30 p.m. Not necessarily. I mean, there's going to be the possibility of, this is the online flexible modality. We're not supposed to have, required meeting times in this online, modality that we're doing this semester. But just so as you know, I'll probably be recording lectures like this, basically like we're doing right now, Mondays and Wednesdays at 8:30 p.m. with the possibility, like if I've got something else going on, if I don't feel well or whatever, that may not happen. But most likely that's going to be the pattern. And that Zoom link is on Canvas. So it's under the, I know it works now. So that's under the lecture module on Canvas, right there at the top. So it's a recurring Zoom link, and you can use that anytime. In fact, you don't even need to have me on there. If you want to use that for study sessions, I think I've mentioned before, if you want to organize a study session with other folks and interact live and chat about course materials, whatever the case may be, you can use that. I do not need to be present. I think I have the settings correct where I, the host, don't need to be present for people to join in. Anyway, going forward, 8:30 p.m. Mondays and Wednesdays. So where I was earlier, I came down from the dean. We had a faculty meeting last week. I was initially going to leave exams, the window of availability for exams for about 24 hours. I'm released at midnight and due the following midnight. But the policy, as it turns out, is a little bit stricter than that. And so I've been as liberal as I can in designating a 3-hour window for the exams. And so I felt like between 5:30 and 8:30 were the times that were most fair. I know a lot of folks work. I myself work. So the exams, I usually budget, for midterm exams at least, they should take you about an hour to an hour and a half at max. And that's if you're a slow test taker. Some people finish them more quickly. So 3 hours is plenty of time. You could start at 7:30 and be done at 8:30. That's kind of, I wouldn't risk it. But anyway, that is the available window, and this is all going to be on Canvas. Just to put it on your radar, the first exam is 3 weeks from today. So that's still, I know, quite a ways away. I know we just started, but please plan on being available during that window for each of the exams. If you have some kind of extenuating circumstance, like if you have a work commitment, if you work in the evenings, send me an email and I'll do my best to make accommodations. I might need to ask for a letter or whatever, but generally speaking, that's the window. And I'm also thinking that's probably going to be the window for study sessions as well, whatever the date is that study sessions are. Anyway, shortly we're going to be doing a recap on Introduction to Ecology. For those folks who are here on the recording, feel free to chime in. I won't pressure you to chime in, but if you want to chime in for those questions, it's basically just a chance to rehash what we talked about in the last lecture session. Then we'll delve into population ecology for the main lecture for this evening. And then I'll do Lab 2 overview with a little bit of review. Okay. So for the folks listening, if there's anything I'm saying that doesn't make sense, please let me know. Again, feel free to chime in at any point. Okay, so for a recap, what we talked about last time, if you remember last time we started at Unit 1, which is the ecological unit of the course, and we introduced ecology, and then we briefly went into population ecology. So just to recap what we talked about last time, I just have a series of questions. First question, what does ecology mean, literally? Well, there's a number of different things it does mean, but I guess I'm asking this in the literal sense. Is it the study of the environment? Is it the study of plants and animals? Is it the study of the house? Study of eco-friendly lifestyles? Well, literally, ecology is the study of the house. But just to emphasize it, it doesn't mean we study houses. Like the more nuanced, ecology, oikosology literally means the study of the house, but in a more nuanced definition, ecology is the study of how organisms relate to their environment and to each other. So it's the study of biotic interactions, but also the study of abiotic interactions. So things that are not living, things like rain and temperature and humidity and all the various things that can influence how life goes about life. So it's a pretty complicated science, and it's got a variety of different tiers that we're just going to explore briefly today, being population ecology. Question 2, what constitutes an ecological population? Instead of reading them out, I'll just go ahead and posit to you the definition that gave last time, which was a population, an ecological population, is a group of the same species in the same place at the same time. Okay, so those three things have to be in play in order for something to be considered an ecological population. If one of those three is missing, it's not a population, it's something else. Okay, so this is again, kind of a breakdown, a different picture, but the same breakdown of the different tiers of ecology. We're looking at this tier right here when we're speaking of population. So an ecological population is a group of the same species in the same place at the same time. Hey, just quick question. And my worst fear is that this isn't that you can't hear me. Amanda, can you hear me? Amanda. Okay, good. Thank you. I appreciate that. I've recorded lectures before and then realized I didn't hit the recording button. It's like, whoa, I don't want to do that. So anyway, thank you for confirming. Okay, so a group of the same species in the same place at the same time, that is what constitutes an ecological population. Question 3, what are the two core concepts of population ecology? This is not the actual ecology of populations, but the discipline of population ecologists, what geeks like me do. Okay, so what are the two core concepts that population ecologists work with? Instead of reading them out, because that's a bit exhaustive and I want to go kind of quickly today, I'll just posit to you, as we talked about last time, that demography and density are the two things that population ecologists are primarily concerned with. So demography meaning things like the sex ratios, ratios of birth versus death, age makeup of a population, and then density is literally the number of individuals per unit square, whatever the unit is available. So just for an example, this is an actual overview of Santa Cruz Island. So if you've ever been to Santa Barbara and you're looking out towards the ocean there, and you see that big landmass off in the distance in Santa Barbara, that is Santa Cruz Island. It's the largest of the Channel Islands. Really cool place. That's where I fell in love with botany, actually. Really, really amazing place. And there are these cute little foxes over there called the Channel Island foxes. And there aren't that many of them, a few thousand. It's actually a kind of a privileged position for folks who study Channel Island foxes and the examples we talked about last week, where you can actually know the exact number of individuals you work with within a certain degree of certainty. And within an island, it's not landlocked, and so it's a very definite piece of land that how much space is available, and you can know exactly how many you're dealing with. So you can know the population demography and the density really well if you're talking about an island, because it's locked, right? If you're working in studying a population that's in the continental U.S., for example, like blackbirds. I think I talked about blackbirds last time. You're pretty much stuck with doing estimates, and very large estimates at that. And I think I also talked about Darwin's finches, looking at demography, and we'll justify a little bit later today why it's important to understand aspects of demography and density. We talked a little bit about them last time, but I digress. Question 4, define the ecological meaning of life history strategy. I will just go ahead and define it as a life history is a set of traits that affect the population's success at reproduction and survival, and that can be many, many things. So that's opening up a whole can of worms. We'll talk about that at length actually throughout the course. Life history is not just something that's contained to ecology. It's really a biological wide a definition that we work with. So broadly, it's a set of traits that affect a population's success at reproduction and survival. And that can be, as I said, many, many things. And then last question, let's distinguish between what we call R and K selected species, which are sort of different ends of a continuum of life history strategies. Remember, life history strategies being this. Okay, so these are different ends of the continuum. We define R selected species as species that have many offspring, little parental care, tend to have shorter lifespans, etc. On the opposite end of the continuum, are K-selected species that have fewer offspring, much more care, and tend to live longer. So it's really a continuum. You know, this is a phenomenon that we'll continue to hit on throughout the rest of the course. Is that it's tempting to want to put things into neat little categories. So, you know, like, you know, every time you go out and look at some living organism, you know, you might want to say, oh, that's R selected or that's K selected. Well, that's not that easy, especially when you're talking about studying life. Life avoids categories. You know, these are just this continuum that I'm proposing to you is just our attempt to try to get a handle on life histories. So it's very much an artificial designation, and I'll talk about that a little bit later on. But broadly speaking, this continuum of R versus K, short-lived, long-lived, many offspring, few offspring, etc. Okay, so with that, we'll just go ahead and carry on with population ecology, really delving into it, the application aspect of it today. This is still in chapter 19 of your textbook, at least in the 5th edition. So just to rehash, ecology literally means the study of the house, oikosology from the original Greek, broken into increasingly complex levels that we're going to explore in varying degrees. Today, looking at populations. Okay, and this is the same schematic that you'll probably tire of seeing throughout the semester, but it's nevertheless important to look at it every chance we get because it's our schematic. And we're starting here with Unit 1, the ecological end of biology, and then we're gonna you know, do a full circle as the semester goes on. But today we're talking specifically about where the red arrow is pointing, population ecology. Okay. And you know, this is literally just what I was talking about a couple slides ago, life history strategies. It's really tempting, you know, once you, oh, R, K, you know, that makes sense. I like it. And you know, it is handy. It's useful to try to get a handle on life because life is so complex. But my suggestion to you is to avoid thinking about things in those strict binary terms because life, as I said, avoids categorization. It's much more of a continuum than anything else. And what works best, the life history strategy that works best for each individual population, each individual species and so forth, doesn't necessarily, you know, exist on a fixed spot along that continuum. Definitely not in these two categories. So it's continuum. It's the many shades of gray, all right, between, and where a population exists in between these two, well, it might be here for this population of the same species, but it might be way over here for a different population of the same species. It just depends. There's a lot of different factors that can affect a population's life history. Okay, and we'll go through some examples, but just to emphasize that this R versus K continuum, it's a continuum. It's complex. Okay. Just to name some examples, and as a botanist, I can't help it. It's my bias, perhaps. I'm going to give you some plant examples to start with, just for a few. And the reason why I start with plants isn't just because it's my bias, but because plant life histories vary a lot. It's, you know, a lot of folks tend to think that plants are these kind of simple organisms. Oh, how that can't be further from the truth. Plants are amazingly complex creatures. They're fantastic, and I love them. So let's just go through a very quick jubilee of different life history strategies that plants employ. And maybe, and we'll rehash these, especially when we talk about plants much later in the semester, but maybe this will emphasize, firstly, life history strategies and how much they vary just in terms of life itself, but also how diverse plants are. So I'll go through some examples. First way in which plants can vary in their life history strategy is that they vary tremendously in size. Okay, you know, huge. They're some of the smallest organisms in the earth, and some of the largest organisms in the earth on the earth. So, you know, on the smaller end, this is doveweed, Croton setiger. Actually, no, I think they changed it. Don't mark me on the Latin binomial there. A doveweed is a common name. If you have a pond in your yard, or you ever see a pond somewhere with ducks and so forth, you might call this that scum stuff that tends to float on the surface. That's this. Each individual little capsule here is actually a mature plant. They're flowering plants. So they're little tiny small flowers. But each one of these things is an individual. And there are my fingers there. So you can see how small they are. That's a mature plant. Okay, they're not seeds. Those are actually the full-bodied plants. Here on the same end, there's my figure again. This is a Tejon Ranch plant. This is Cambia candida, which is white pygmy poppy. These are actually poppies, very closely related to our state poppy, Schultzia californica. So those orange plants that you may be familiar with. Actually, it's on my shirt today. I didn't realize that, but actually, if you can see my shirt, that's our state poppy. This is in the same genus right here, but obviously much smaller. And then, okay, so these are some small examples, but then on the opposite, so you might call these R-selected, right? On the opposite end of the continuum are some extremely large plants. And so look at this. This is kind of like taken from a helicopter, and it's actually not my photo. Actually, frankly, I forget whether this is taken in Colorado or Switzerland, because this phenomenon exists in both places. But let's just call it, they're both aspens in either example. But there's this aspen right here, and I say singular aspen because you can imagine this is a landscape photo, but this is all one plant. Okay, it looks like a forest at this distance, doesn't it? But this is all one single individual over a square mile, and it's all one plant that all shares the same root system. So functionally speaking, it's the same exact individual. Okay, so a huge plant in the singular. Okay. And then maybe a little bit closer to home, maybe you've seen this before, the General Sherman tree, which is a giant sequoia right here. You can go see it next weekend if you want. I think the national parks are open. It's just about 2 hours northeast of Visalia. So really, it's really not so far here from Bakersfield. This is the largest organism on planet Earth, single organism, not colonial, but the largest single organism, by volume in the world. Okay, so this is a huge tree. Ironically, its seeds are tiny, and I'll show you a picture of its seeds in a couple slides. But this is you look it up, General Sherman with person at base on Google, and a mature person, is like just a little bit taller than this fence, and this picture doesn't even do it justice. This is a huge tree. Okay, so variation in size, variation in lifespan. So, you get things like Hirschfeldia icona, which is a short pod mustard right here. It lives for about a week. It germinates, it grows really quickly, it produces seed, and it dies. It's an annual plant. Same thing with this Ericameria here. Forget the common name, but it's a tiny little thing, about 1 centimeter tall. It germinates, it produces flowers. Those flowers get pollinated, and the adult dies, the seeds go back into the ground. Lives for about a week, 2 weeks at most. And that's it. Okay, so certainly R selected. On the opposite end of the continuum, the K selected. These are both actually California examples. If you've ever driven toward, Highway 14 on the other end of Tehachapi, or you've driven to Las Vegas, or through Utah, Arizona, you've seen this. This is creosote shrub. So that the Mojave Desert. These are actually some of the oldest clonal species in the world. They don't look like it, but these things can live on the order of 2 or 3. I think the oldest documented individual is like 4,000 or 5,000 years old. These are old shrubs, clonal, okay? Kind of like those aspens in the last slide. So they're not individuals in the pure sense, but they're still really long-lived plants. And then also something that you can go see on a weekend, this is the Methuselah tree. It's a bristlecone pine. It's the oldest individual organism in the world. In terms of the individual, and I forget the exact date, over 4,000 years old. I think 4,800 something years old. Really old, and as old as ancient Egypt, right? And it's still living. Okay, so its leaves are older than some folks in the class. I mean, its leaves can live for over 30 years. So it's a really old organism. Really cool, certainly on the K selected end of the continuum. Plants can also vary widely in their dormancy. So things like this sunflower here, another California native, Helianthus annuus. It's an annual plant, and it basically just grows once in a growing season, and then it dies back, and it comes back the next year very consistently. Things like onions and garlic, bulby type plants, same thing. They're perennials, but they go dormant in the winter, and the bulbs maintain the energy under the ground in the winter, and then they grow back the next year. If you have spent any time in the Midwest or the East Coast, the great deciduous forests, you'll recognize they go dormant in the winter, but then they come back the next year. Okay, so in terms of dormancy, their dormant periods are far shorter. So in that sense, these species here on top, they are certainly more on the R selected end. But the species here on the bottom, a bit different. This right here is a picture of a resurrection fern. It's the common name, resurrection fern, and what's one here on the left actually looks dead. It can be completely desiccated cells for on the order of years. I think the oldest known example of this, there was a Stanford professor of botany, Hal Mooney, who's still living. He taught at Stanford for like 50 years, and he had a resurrection fern in the back of his lab that he had never touched. He'd inherited it from his predecessor in like the 1960s and never touched it. And then when Hal Mooney retired, they were cleaning out his old lab, and they found these resurrection ferns dated from like the 1940s, and they knew they were resurrection ferns, so they stuck them in water, and lo and behold, they rehydrated their cells, and they looked like this within a couple days. So they can be dormant for a very long period of time and be just fine. Pistachios are, well, not quite as an extreme of an example, but pistachios can, you ever drive past a pistachio orchard that looks like it's been dead, and you drive past all the time, like it's been dead for years? Well, pistachios can be dormant for, I'm not a pistachio expert, but they can be dormant for a few years before they resprout again. It just depends. Depends on the way the farmer operates them. Anyway, they can be dormant for multiple years before resprouting. hard crop to grow. Variation in growth rate. So here are the R-selective species on the left and the K-selective on the right. Bamboo is one of the fastest growing plants in the world. It grows between 7 and 10 centimeters a day. So you can stick a camera in front of it, and at the very end of the apical meristems, and just set it on record, and you can watch it grow within the course of a day. If you're really patient, you can, you can't see it with your naked eyes, but you can literally watch it grow. It happens pretty quickly. And this is, I forget, this fan palm here, Southeast Asia native, same thing. It grows really quickly, 5+ centimeters a day. So, very fast growth rate versus things like Monterey cypress, which is another California conifer. This is a several 100-year-old plant, but it's not that tall. But a human adult would be, this is about a 10-meter tall tree maybe, so a human adult would be somewhere right here. It grows very, very slowly, like a couple millimeters a year, if that. That's an ideal year. And then, not truly a natural example, but the bonsai trees of, there's bonsai trees in Japan that people have been taking care of for 500 or 600 years, and they, you're taller than them. I mean, this is a big example. I mean, it's on a table, but it fits inside, but a few hundred years old, but grows very, very slowly. I digress. Variation in fecundity, which fecundity literally means the number of offspring. Okay, there's a little thing about this in terms of the number of seeds for plants. So on these examples here on the left, these two, actually I forget what they're called because they're not my pictures. Actually, I try to use my pictures when possible, but these aren't. Thousands of seeds produced in the growing season of every year, versus, actually this is my picture, Picaringia montana, which is chaparral pea. It produces, it's a perennial plant. It produces maybe 6 or 7 seeds every few years. so really very few. This is a calcornis, which is a Mariposa lily, produces one seed every year, maybe. And then actually, I talked about this last time, the carcass plant, which is, that's a single flower there. I think it's in the same family as avocado. So think about the avocado fruit, one big seed inside. The thing about, there's a good size comparison, actually. There's a standard size calculator, so that gives you an idea about how big this flower is, a big flower, and produces one seed, and it only flowers like once every century, 60, if it flowers every 60 years or so, that's a pretty good rate. So variation in fecundity, R selected versus K selected. And then I think this is the last one. Oh, second to last one. Variation in seed size. So small seeds to large seeds. Remember General Sherman, the largest individual organism in the world? These are its seeds. So, think about that. These tiny little seeds can grow up to be something that's so enormous. It's pretty amazing. These are poppies, poppy seeds. You'll see them on your bagels. This is actually the same genus that people have fought wars over for opium. I digress. I won't go there. Anyway, pretty small seeds. And then on the opposite end of the continuum, obviously coconuts. Each individual coconut is a seed. Okay, it's not a fruit with many seeds inside of it. It is a single seed. And then avocados, one single seed inside. Excuse me. And then this is the last one, I promise, I think. Yes, last one. The variation in the number of reproductive bouts. So these two here on the left, this is a Clarkia named after the Clark, the Lewis and Clark expeditions. These species here will reproduce, many times within a single growing season. So from like January to March in a given year, it might, germinate and produce seeds, and the seeds will fall to the ground. Those seeds will germinate, produce flowers, and produce seeds. And grow, it'll do that, like sometimes 5 or 6 times within a single growing season. So, many reproductive bouts within a given year. Whereas on the opposite end, I'm just pointing to my favorite pictures right here. This is the century plant. It's in the agave family. You've probably seen it before. The common name suggests it only sends up an inflorescence, which is a flower stalk, about every century. And so, that's it. For the rest of the time, once it does that, the base dies. So, for most of its life, it looks like this. It hasn't even reached sexual maturity yet. And then things like valley oaks, which is the largest oak species by canopy volume. We've got a lot of those on Tejon Ranch. It reproduces, it produces a lot of acorns in a given year, but very rarely will one of those acorns actually sprout. And so a very, very low rate of reproduction. I digress. There's so many pictures. I have to stop myself here. Okay, so that's just what I hope was a quick survey of the diversity of how not just plants, but how life itself can vary in terms of their life history traits. And there are many, many more examples. Those are just a quick few. Right now, I'm just going to switch for the rest of the lecture today to how do population ecologists actually study populations. I'm going to posit to you 2 primary ways that they do that. The first is modeling, which is an indirect method. And the second is field surveys, which is a direct method. Okay, so ecologists do both. They indirectly study populations and they directly study populations. And there's me on Tejon Ranch last year, 2019, in what was probably the best poppy field in the state. And maybe, even though Tejon didn't do wildflower tours this year because of the coronavirus, maybe next year we'll be able to do tours. If that looks like fun to you, like Wizard of Oz fun, if you've watched the movie, you'll get that. Check it out. Tejon is awesome. So the first way that population ecologists study populations is an indirect method with the use of models. Firstly, let's define what a model is, because that's kind of one of those colloquial words that people use for different ways. in terms of ecology, well, firstly, in a scientific way, what is a model? Well, a model is simply an idealized version of something based upon certain assumptions. And so meteorologists, people who predict the weather, they have a fairly complex algorithm that allows them to predict the weather. That's a model, okay? It's just an idealized version based on past collected data that allows them to predict to a certain degree what might happen in the future. So it's an idealized version, okay? If you make certain assumptions, it'll give you predictive power, okay? If you have a set of data from the past and you take that data and you work with it a little bit, make an algorithm or do whatever, and then you attach certain assumptions to that data, you might be able to predict what will happen in the future, okay? So if you apply a model, which is simply an idealized version of something for predictive purposes, if you apply models to ecology, to populations, you can do a couple of different things, okay? Firstly, remember, models are based on assumptions. So firstly, if you assume that a population that you're interested in studying has access to an infinite amount of resources, which I know isn't true, right? There's no population that has access to an infinite amount of resources. And think of resources in terms of things like water, things of like energy, space, even time, that's a resource. So if you assume a population has access to an infinite amount of resources, which I know, again, isn't true, but it can still be helpful to idealize such a population. Let's just go ahead and call that kind of model an exponential population growth model, okay? And this can be useful. This idealized version of a population, it can be useful to try to understand what's going on there in certain circumstances, like after a fire. I'll show you a picture in the next slide. Maybe it'll help solidify it a little bit. Versus what is definitely the case for pretty much all populations in a realistic sense is that they have limited resources. So assuming a population has limited resources, that's a logistic growth model, okay? So that's talking about the various ways, analyzing the various ways a population might be restricted in terms of its life history. And from that, if you talk about limited resources, whatever that limited resource may be, it varies by population. If you're talking about limited resources, I'm going to posit to you, sorry I use the term posit so much, but I'm going to posit to you a really important term. So take note of this. The carrying capacity, a really important term. So carrying capacity is literally the maximum number of individuals that a population can sustain given its limiting factors. And we'll run through a number of examples of that. But carrying capacity, in the context of a logistic population growth model, it's the maximum number of individuals a population can sustain with its limiting factors. And that can be very useful. If you have predictive power over the maximum number of individuals that a population can sustain, that's huge, right? Not just from kind of a plant and animal perspective, but demographers, people demographers, and sociologists and agronomists use this all the time. To talk about what's the maximum number of people we can feed with these circumstances, with this amount of water, and so forth. It's a very relevant term. It's kind of the to be or not to be question in terms of a lot of things. What's the maximum number? Well, that's the carrying capacity. Okay, I'm going to keep it to the plants and animals, of course, because we're focusing on really the bread and butter of population ecology, but this can be wider to humans as well. Just saying. So here are just a couple of figures talking about these two different models that we talked about. Firstly, the exponential model, which assumes unlimited resources, and the logistic model, which assumes limited resources. So in an exponential model with unlimited resource, population size here on the y-axis, and time here on the x-axis, As you can imagine, it's just exponential. It just keeps going up and up and up. The population just keeps growing over time because there's nothing inhibiting it at all. Okay, but if you throw limited resources, whatever the limiting resource may be, into the context of a population, as you can imagine, there's going to be a growth period, but eventually there's going to be a point where that plateaus because it's reached the maximum that that population can sustain. That's your carrying capacity right there. Okay, so that's logistic. Logistic population models can give you an idea of where that carrying capacity is, right? So just for some relevant picture examples, why would you even want to know what an exponential, why is an exponential population growth model useful? Well, in certain circumstances, that can actually be useful. One of them is actually really relevant here in California. This is over in the Inyo National Forest a couple years ago. About a 10,000 acre fire burned through, killed all the trees, all the above ground vegetation. It basically looks like a moonscape, doesn't it? When a fire burns through an area and it kills all the above ground vegetation, kills all the herbaceous plants underneath, it looks kind of dead, doesn't it? But there's a lot of potential there. All the carbon, all the nitrogen that was composed there, it basically just got burned out. A lot of it goes right back down here in the form of ash into the soil. And you can't really tell in this summertime picture, but when the rain comes along and gets the soil wet, all the seeds that are alive and healthy in the soil, they get some water in there, and all that extra nitrogen, all that extra carbon, all the extra micronutrients from that fire, in the spring, this can be completely different. For the sake of that short period of time, those little plants that germinate immediately afterwards basically have access to unlimited resources. Okay, so in certain circumstances, like after a fire, think about this picture, that the immediately succeeding things that come in next, those basically have access to unlimited resources. And so an exponential population growth model can be really useful under those circumstances. And there's a variety of different ways that can be applied. Versus a logistic growth model, of course, which is more fitting for the vast majority of populations. Think of things like this. Okay, this is actually a Tejon Ranch picture. Not the best picture, but nevertheless fitting. These are actually little lupin plants, which are in the pea family. Okay, if you're a population ecologist that wants to study this little population of lupins here, okay, and I'll justify why lupins are so important. We depend on them, you and I, for existence. I'll talk about that later in another lecture. I digress. If you want to understand how these lupins are performing, you can use a logistic growth model. And what are some of the limiting factors that are influencing this population? Well, a number of them. You can think about in terms of water, in terms of time to grow. But one that's really obvious right here in the picture itself is that these lupins are co-occurring, competing with these annual grasses here, which are actually not native. So this is avena, it's an oat. These down here are brome grass. They are competing for water and resources, nutrients in the soil. So that is certainly a limiting factor for these lupins here. If you have some past data and you make some assumptions, a logistic model can help you understand how these lupins are going to perform over time. I digress. Just another picture of carrying capacity. The red line here represents exponential growth. The blue line here represents logistic growth. That carrying capacity is basically represented here by this dotted line. So logistic growth models, where it plateaus, that's your carrying capacity right there. And literally the number of individuals per time. That's a very common way that population ecologists plot and track populations. Okay, so I've posited to you method number one that population ecologists keep track of populations, which is an indirect method using models. The second is a bit more intuitive because it's direct. It's literally going out to the population and looking at it and measuring it, collecting real data, okay, live data. So plant biologists like myself, for example, often will go to a spot over the course of many years and will stake what's called a transect. So like stakes here. Okay, so stake and then stake at the other end. Let's call this a 100-meter long transect, and then run a tape measure between these 2 points. And you're measuring the exact same stretch of tape in space over many years. And so this is actually the same exact transect, sometime in the 90s, sometime in the early 2000s, and sometime, well, 2002, 2003, and this is sometime in the 90s. These aren't my pictures, these are one of my colleagues' pictures, but I digress. Same exact transect over time, and that's just direct field survey data. But because I've been talking so much about plants, I thought, you know, and I've done this the past couple of semesters, I'll talk about these direct field surveys in terms of wildlife, because they're perhaps a bit more relatable, and wildlife are awesome too. So, you know, a lot of wildlife biologists have studied wildlife directly through most time through the very old school technique of stalking. Okay, so, you know, if anyone is a hunter or has known someone who hunts avidly, stalking is the term for following animals so that you can, of course, hunt them. But if you're a wildlife biologist and you are just wanting to collect data on a given species, not to hunt them, but just to collect data, you're stalking them as well. You know, stalking is another one of those words, but, you know, in terms of population ecology, wildlife biology, you know, tracking an animal and directly collecting data on them, that's stalking. That's through most of the discipline of wildlife biology, wildlife population ecology. That is how they've collected data. And that's really hard to do. If you know any hunters, ask them how hard it is to track an elk. It's really hard to do. That's probably one of the reasons why I went into plants, because plants don't run away from you. Okay, now, so it's hard to stalk, right? So thankfully, in the last really the last 10 years, the field of wildlife biology has become a bit more sophisticated. It's gone away from stalking because it's invasive. It you know can scare animals, and you know, you're not necessarily seeing the most natural behavior, because once you see an animal, they often run away from you. Camera trapping. Well, actually, no. Sorry, I'm getting a little ahead of myself here. Stalking is hard. You have to be really skillful or lucky to come across wildlife. And these are actually all my pictures from Tejon Ranch. I was lucky to get pretty close to a bear one day within a few feet of my truck. Big, you know, big scary male black bear. These are actually pronghorn antelope over on the Antelope Valley side of Tejon Ranch. A dead barn owl, so that's about as close as I've come to an owl on Tejon Ranch. They're pretty hard to get close to. A red-tailed hawk with a broken wing, which I backpacked it out and took it to a rehabilitation center. I think it lived. So that was the closest I ever got to a red-tailed hawk in the wild. Usually they're soaring way above you, right? You know, so pretty. You know, cattle. When cattle are first born, they're a little bit stupid, and they often will run right up to you and lay down in front of your car, and you have to actually get out of your car and pick them up and put them on the side because they think the car is mama. So cattle, sometimes wildlife will run right up to you, or if you're studying amphibians or so forth, you know, you might be able to get lucky and catch them with a net for studying purposes. I digress. No matter what form it takes, no matter what taxa of wildlife you're studying, it's always hard. You have to be skillful and lucky in order to get good data using stalking. But as I said, camera trapping is a more progressive approach to studying wildlife. And the video footage that I actually showed you on the very first day, check out the first lecture, that Ben and I, one of my colleagues at the Conservancy, put together. That's all camera trap footage. And so it's an alternative to stalking. It basically involves strategically placing motion activated cameras along a water source or along a game trail in a field of view that will actually capture footage of wildlife. There's no harm to humans or animals involved. It's a lot less work because all you got to do is place the camera and it does all the work for you. You get a lot more data, and the data tends to be higher quality because you're getting behaviors that you probably wouldn't see if you were there, because the animals would be scared of you, right? So camera trapping, it's an alternative to stalking. It's the only way I've ever studied wildlife, because I don't like trying to find them. It's hard. This is the easy way and the better way. So you can, with that, you can get things like this. This is obviously not from California. This is a snow leopard. It's the rarest feline in the world. Most think of like Siberia. It's thought that there's less than 100 individuals left in the wild. Actually, there are a couple at the Santa Barbara Zoo. There's a lot in captivity, but it's the rarest. It's the rarest feline in the world up until. If you've ever seen Planet Earth, the documentary, a very groundbreaking documentary on, I recommend it. It's probably still on Netflix. That they use camera traps to get some of their footage. And up until about 10 years ago, Planet Earth was the one who did it. They actually didn't have footage of a snow leopard. People had only ever studied it from way far away. Indirect, precious few photos, so with the advent of wildlife camera trapping, it was some of the first close-up images and the first close-up footage that had ever been taken of this really rare feline species. So there it is, beautiful imagery, right? And you can study this and really get an idea of the population ecology of snow leopards, okay? Another cool example, imagine trying to be a person climbing that tree way up in the canopy of the Southeast Malaysian rainforest and being so still that this orangutan wouldn't be freaked out by your presence. You couldn't do this as a person. This camera trap was able to capture this amazing footage of this orangutan. Pretty cool. The problem with camera trapping, though, is that it's too good. You tend to get your data source is basically photos, right? Usually it's photos, not videos. You get millions of photos over time. And it's really labor intensive to process photos. You literally think about it, having to go through them one by one by one and collecting data on the basis of what the visual cues tell you. That was actually one of my first projects. I actually interned at Tahone Conservancy before I got a job there. And that was one of my first projects, was literally going through picture after picture after picture and looking at various demographic and density data from these camera traps, looking at pigs on Tahone Ranch mostly. So that's the problem. It's really labor intensive to process the data on the other end. But the field has gotten even more progressive since then in the last few years. In 2017, a paper was published, which the Tahone Conservancy, actually it's 2018, which the Tahone Conservancy collaborated on, where we had this grid of 48 cameras out on Tahone Ranch, mostly looking at the population estimates of feral pigs on Tahone Ranch. The overall goal of this APHIS project was to collect millions upon millions of photos, not just of pigs, but of basically all wildlife, in order to teach a machine how to recognize species by the pixel information without even having a person look at a picture, literally plugging the JPEGs, the raw images, into this computer, this algorithm that the math goes way above my head, a variety of inputs and so forth, functions. The output basically is, this picture is this species, and there are that many individuals in this picture, and it basically does it automatically. It's a model, right? It's basically, taking data and applying certain assumptions to it with mathematical functions and giving you some really robust results. So I'll give you some examples about how this works. It's really interesting. I can distinguish, this is actually really powerful. I mean, I think the machine learning, the model is way above 90% accuracy at this point. So it's really awesome. So you can imagine you have millions of photos from a camera trap array, and you don't know what to do with them because it's just way too many photos to process. You can't pay enough interns to do it. It's just a lot of photos. So you come up with this, you give all your photos to this team that has developed this machine learning software, and they plug it into their software, and it can do things like, okay, without even looking at the photos, no humans involved, these are some pigs. That's not a pig. It sounds kind of simple, but think about it. It's a machine that's doing it. It's a computer program that's identifying that that's a pig very accurately. It can be even more sophisticated than that. It can identify, of course, things by species. So plugging images into the software, this is a bobcat, right? A machine can do that. Bobcat, mountain lion, and then even identification by individuals. So even more than by species, it can differentiate different individuals of the same species. And so let's say this is individual A of this mountain lion population, but then it might recognize other individuals within the population. Identification of parents and offspring. And so the machine can actually pick up, even by this kind of crappy photo, this is the parent and this is the offspring by pattern coloration of these mule deer, okay? Pretty handy. And imagine this again in the context of millions of photos, okay? So you're getting lots of data. Identification of parents and offspring, a couple of adult sows here with their little cute piglets. Actually, they're invasive, so they're really nasty. I digress. But really handy, and it can give you really robust data, how many individuals there are. This is Mother A from, she's 10 years old now. She produces a clutch of X number of piglets per year and so forth. So think about it in terms of, again, having many photos over a long period of time, what this can tell you in terms of the demography of a species and the density of a species, of a population rather. Parents and offspring again, so these, in this case, gray foxes with a couple of, cute little fox cubs. Parents and offspring again, this can recognize by individuals, so mama lion and her 3 cubs. Parents and offspring, mama skunk and, cute little baby skunks, right? Parents and offspring, mama bear versus cute little cubs. Oh, they're so cute. If you ever see a bear in the wild, if it's a male walking by himself, a lot less reason to be scared. The adult males are cowards usually. The dangerous ones are the females, especially if she has cubs. Just a word if you're ever out hiking in nature. If you see cubs around, that's the most dangerous situation to be in around bears. Parents and offspring. So, I mean, look at, this is a bobcat carrying a little cub in its mouth, as cats are wont to do. The machine learning software is sophisticated enough to the point that it recognizes that those are 2 individuals. That's pretty handy. That's pretty sophisticated technology right there. Identification by gender. So these are all adult mountain lions, but it can recognize, this is a male mountain lion and these are 2 female mountain lions. And it can, again, recognize by individual. Identification by species, and so the machine learning software might look at this and say, you got a skunk and a gray fox in this same image. Okay, so those are just some examples. Imagine that again in the context of millions of photos, and you're just taking those millions of photos and dumping them into this pretty sophisticated software, and it's giving you robust, analyzable data with relatively little work on your end. So it's revolutionized the field of wildlife biology. However, there are a few drawbacks to camera trapping. The one is that you're leaving these cameras out in the wild by themselves for long periods of time. Therefore, they are subject to things like vandalism. So people will, there's been instances on the ranch of people stealing cameras, and these are expensive cameras, on the order of like $2,000 or $3,000 per camera. Okay. Even animal vandalism. So a bear might come along and completely bust open a camera cage right here, exposing the camera itself. This is a picture of a camera that was buried under a mudslide. Okay, so obviously that did that camera in. And another camera that was just placed a little bit too close to a creek, and the winter wet season came in and it flooded the camera and ruined the camera. Okay, so there's just some drawbacks. Okay. So we've talked about modeling and we've talked about direct surveys. Those are 2 methods that wildlife or population ecologists more broadly have studied populations. Why do this? What's the point in population ecology? Well, there's a bunch of different ways, and I'll continue to justify this throughout the class. But just for a few quick examples, I'm mostly going to tie this back to where I work in terms of Tejon Ranch, and I really wish that we were an in-person class so I could invite you out to Tejon Ranch maybe later. But conservation is a huge thing. Maybe you've already noticed some pragmatic, applicable things that you can do. If you have robust population data, you can use that data to manage the populations that you're studying, that you're working with, for a variety of different ways. Think about like endangered species and even things like you don't want, like invasive species management. On the opposite end, things that you don't want around, you can use population ecology to get a better idea of their fecundity and so forth, the number of individuals they produce, yada, yada, yada. Protect the species, I'll show you an example of this on the next slide. Resource management, and a huge thing, this is kind of becoming a key word here in the last few years in the discipline of population ecology, is ecological services. So that's an important term. It's basically a term that refers to the services that we humans get as a result of healthy, intact, functioning populations and ecosystems. So it's one of those things that is hard to quantify. There's every 10 years, there's this group called the Millennium Ecosystem Assessment that tries to put a numeric value, an economic value on things like clean air and clean water and so forth. Ecosystem services, things that human beings derive out of functioning ecology. And they've estimated it on the order of $33 trillion annually. All the medical expenses that are saved by clean air and clean water, and go down the list. Okay, they've estimated that. I don't know how exactly they get to 33 trillion, but 33 trillion is more than double the GDP of all world governments as of 10 years ago, at least, which is the last time. It's actually 2020 now, so the Millennium Ecosystem Assessment should be coming out again sometime soon. I'll be interested to see what the most recent estimate is, but as of 2010, $33 trillion, which is a heck of a lot of money from ecological services. I digress. So just some pictures. This is a tamarisk tree on Tejon Ranch, which is an invasive species that we monitor the population ecology of them fairly robustly because they are really nasty. They steal water from adjacent plants, and they do things like reduce berry habitat, reduce amphibian habitat. They actually are even bad for the cattle, so the ranchers don't like them because they ruin the water table of the creeks that they grow in. So we've used population models and even direct surveys to get an idea of their fecundity over time. And some of the data we've collected on that has allowed us to make some management decisions, like releasing these beetles that are their natural predators as a biocontrol. These are Diarrhobda beetles, tamarisk beetles, that we release, trying to basically eat the tamarisk trees. And this has actually been fairly successful. I don't remember what I put this picture in here for, so I'm going to skip it. California condor. I'll just go ahead and say this is the high country of Tejon Ranch. So that's looking actually, over on the other end of this ridge here is, dip down your, looking north toward Bakersfield. So that, that's a good picture of the high country of Tejon. I digress. I can't remember why I put that in there. California condor. I think I talked a little bit about that last time. At one point in the 1970s, there were fewer than 30 individuals left in the entire species. It's thanks to population ecologists for studying them and taking quick action to save the species. You'll see a lot of condors on Tejon Ranch up here in the high country. So models as well as direct surveys. And then this is a plant example. It doesn't look like much, but this is the Tehachapi buckwheat, which is Eriogonum callistum. It's completely endemic to Tejon Ranch. So it doesn't occur anywhere else in the world. That species, it's a pretty remarkable species, and I just love it. It's another one of those rare things. It's kind of the plant counterpart to the condor. Handy to understand its population ecology. Okay. Oh, critical. This is just a picture. Here's Tejon Ranch here on the right, just to show. Tejon Ranch is basically at ground zero for critical California condor habitat. So the population ecology on Tejon Ranch of condors is pretty robust, and it's one of the key things that we do. Anyway, I digress. So that's it for lecture for today. I just really quickly want to introduce this week's lab activity, which is called Today I Am a Biologist, Part 1. I put an instruction document on Canvas. So that should be there live until week 2. Take a note, that instruction document is just an instructions document. It's not a worksheet. So I'd still like you to do the actual lab in your notebook. So it's a little bit more formulaic than last week's. I've given you specific questions and specific things that you need to do. So as long as you number it and title it in a clear way so I know what section of it you're working on, that's what I'd like you to do. So anyway, take a look at that document. The broad focus of this lab is basically reiterating the scientific method. So I've given you 2 practice scenarios, basically 2 situations that paragraph, describing some kind of situation where you are going to go through the steps of the scientific method. And then the third thing, and you'll create a graph for those things, and I'll show you some examples. And then the third thing is I ask you to create your own, to run through the scientific method of your own scenario of your choosing. And a good place to start would be whatever you journaled on last week. Think about what you journaled on last week. You were observing a single individual. Now take that and use your imagination to apply that to your own situation where you're devising your own experiment. Okay, so I'm going to go through some review really quickly here because there's a couple things I don't think I hit on very well during the scientific method lecture. But this is going to be due a week from today, Monday, September 7th. Just a quick rehash. These are the same slides from the first lecture. Just a quick rehash of the scientific method. Remember the king snake versus the coral snake example. So the coral snake is the venomous snake. Whoop, what is going on? Sorry about that. The coral snake is venomous. The king snake is allegedly what's going on? Sorry about that. The king snake is the mimic. So remember, the steps of the scientific method are question. I'm not going to rehash all the details. Hypothesis, or observation, then question, then a hypothesis, then a prediction. Right? Remember the experiment that we set up, where my mouse is acting weird. Come on now. Okay. Remember the experiment that we set up, where we had a certain number of snake species in an enclosure, and then we set loose some predatory birds over the enclosure, and basically measured how many of each species or color patterns of the different snakes were predated upon. This was a way to test the hypothesis of whether king snakes received protection from their danger pattern relative to coral snakes. So remember we talked about things like the independent variable and the dependent variable. The independent variable is what's manipulated in the experiment, so it's literally the different color patterns of snake that we chose for the experiment that we put in the enclosure. King snakes, rosy boas, rattlesnakes, whatever the case may be. That's the independent variable. It's what does not change. And the dependent variable is literally what's measured. It's in this case, we measured the different rate of predation of the different snakes. And I know this is a little bit of rehashing. It's the same example that we did in the very first time. So that's the independent variable versus the dependent variable. Important concepts. You'll be asked to apply these same concepts to different scenarios in this week's lab. So independent versus dependent. But one thing that I don't think I really adequately covered in the first lecture is control variables. Okay, so that experiment that we set up, that hypothetical experiment where we had the enclosure and we had the different snakes in the enclosure and the rattlesnake and the red-tailed hawks flying over, predating on them, that was all the same treatment, right? I mean, it was basically death by bird, right? That's the treatment, to test our hypothesis. But there's one thing that I didn't really discuss, that we didn't really do in that hypothetical experiment, which was we didn't control it in any way. in the context of any experiment, it's really bad form if you don't have the same set of whatever it is you're studying, but under conditions that don't change. Okay, that's called your control relative to the treatment. Okay, so in that, going back to that example, a control, what in the world? I'm sorry, please excuse this. Okay, so going back to this example, the control is, let's say you have two enclosures now, two enclosures. One enclosure is one in which the red-tailed hawks are allowed to fly over and eat the snakes. The other is one in which the red-tailed hawks are kept away from, but it's still got the same number of different snakes inside. So it's got the same number of snakes, you know, same conditions, except it doesn't have any bird predation. So no change throughout the course of the experiment. It gives you a baseline, okay? It's important to have that baseline in the context of any experiment in order to, you know, basically call it a fair test, okay? So this can be in many examples. You know, maybe you've heard of placebos before, like for pharmaceutical testing, okay? So if people are testing the effectiveness of a vaccine, like the coronavirus vaccine, this is probably what they're doing right now in trials, is they have their vaccine, but then they randomly administer that to a group of people, randomly selected people, but then they also randomly administer basically this, you know, people don't know what it is, but they administer it basically nothing, like an injection of water, a placebo, and people don't know whether they've gotten the vaccine or the placebo. And then they measure the response, the increase in resistance to the disease or whatever the case may be. That's a control too. A placebo is a control. It's unchanging. Okay, so it's kept constant for a fair test. And I don't think I talked about control variables. I mean, you need that baseline for any kind of fair, unbiased experiment. Okay, so I'll ask you to actually create control variables in this week's lab as well. Okay, and then of course, you make a conclusion, you accept or reject. This is all review. Okay, and so the last thing and I'm talking kind of long now, is that I've also asked you to create a graph for each of these scenarios. Remember, I've asked you to do two scenarios, one which I've kind of laid the groundwork for you a little bit by giving you the scenario. The third, I've asked you to do it yourself. I'm going to ask you to create a graph on the basis of your hypothetical experiment. And so these can take a number of different forms. One option at your disposal is to create a pie chart, a pie graph. And so this is actually what the pie chart for the snake experiment might have looked like. I made this up. This is just for pretend. So, you know, this is what, you know, I think, you know, in the course of that hypothetical experiment, we found that king snakes, which is the gray color here, the king snakes were predated upon at a far lower rate relative to the other snakes that were put inside that enclosure. So we concluded that king snakes actually did receive protection from their color pattern. So we accepted our hypothesis. Remember back to that experiment? Okay, so this is actually a graphical representation of the results data right here. Okay, and so this is what it can look like. Something like this, that's color coded, and then a caption here at the bottom that basically describes what's going on. Another choice is a bar chart. I'm sure you've seen these before in other classes. You know, I've asked you to create a figure for each of these three scenarios, okay? One thing which is really important to point out for these hypothetical experiments is that, I'm gonna exit screen share here. One thing that's important to point out for these hypothetical experiments for these labs is that, you know, you're doing this as part of your biology lab notebook. This is just for pretend, okay? So in the course of this hypothetical exercise, I'm asking you to, you know, basically make up data, make up your results, make up, state your conclusion. It's just, it's like a trial run, it's for practice. There will be labs later on in the semester where I actually ask you to go and collect your own data and really affirm or reject your hypothesis in a real way. This is just kind of pretend. And so I just want to emphasize, and I think I have a little bit of language in, and actually, let me go ahead and share what this document looks like. No, where is it? Right here. So here's the actual document right here. It's never okay to make up data or results. That's an unethical thing to do under normal circumstances. It's like if you were to try to publish this data in a peer-reviewed journal article, that would be a no-no. But for the sake of this lab, you're making up data, and that's fine in this context. I just want to emphasize that so you don't get the wrong idea about what I'm asking you to do. Hey, this guy's asking us to make up data. I'm not. Well, yeah, I am asking you to make up data, but it's for practice purposes only. A little bit later on in the semester, when we've had a little bit more time to talk about the scientific method, I will ask you to collect, to make your own hypothesis and collect real data in a real way, and then create a graph and basically go through this exact same process in these different scenarios, but in a real way, in something that you're not choosing, something that you're not making up. Okay, so it's all fairly self-explanatory. Here's scenario number one, where I've asked you, you want to determine the effect of a certain fertilizer on orchids growing in a greenhouse, and so you have 100 plants to work with, and so I've asked you basically these questions. These are what I want you to answer in your lab notebook, still following the same format of page title, titling the whole, this is the title here, obviously, of the lab, and then, follow through practice scenario number one, question 2, question 3. This isn't the worksheet, this is just instructions. Same thing, practice scenario number 2 with a number of questions associated with it, fairly self-explanatory. But thirdly, I've asked you to devise your own experiment and go through the exact same steps of the scientific method through these questions, okay? And then the last thing, I guess, this is kind of a side, is to write a reflection paragraph or two about what you learned from the lab. So that's at the very end. Just a suggestion, you don't have to do this, but since you already did a lab assignment last week where you looked at an organism, whether it was, I noticed when someone did chickens, someone did grass, and the person did pigs. I just took a quick look at them earlier today, looking good. Feel free to use the organism that you chose and that you observed a little bit in your situation, and now use that as the framework for this hypothetical experiment that you're going to design. In this case, making up data. Okay, so feel free to use any kind of gleanings you have from last week's lab assignment to help you with devising your own experiment for the purposes of this week's lab. Anyway, how do I exit screen share here? Okay, that is it. Morgana, Amanda, do you have any questions? Thanks for listening. You hung in the whole time. Thanks. This is awesome. I realize I'm a chatterbox, but any questions?
None that come to mind right now.
Great. Well, hopefully that means I was clear. If there's anything, feel free to email me at any point. So you thought that was pretty clear? Because this is the first time I've asked a class to do this type of thing. So, I guess I want to make sure that I'm explaining everything. Okay, good. And Morgana says it was clear to me as well. Thank you so much for clarifying, both of you. I really appreciate it. Okay. Well, any other questions before we sign off for this evening?
No, sir.
All right. Well, have a good evening. And for folks who are watching on the recording, this is kind of going to be the normal pattern. You're welcome. This is going to be the normal pattern. I highly encourage you. I'm going to be taking notes of who is attending. I don't promise it. So, but I might be throwing some extra credit points at people who consistently attend these live lectures. Again, this is not required. Yeah, I certainly highly recommend it. And this is going to be our normal pattern. Usually Mondays and Wednesdays at 8:30. Some weeks, I can tell you now, it's not going to always happen this way, but most weeks probably. And they're not always going to be this long. I tend to get chatty about these first lecture units because it's really where my bread and butter is. There's going to be times where we definitely do not talk this long. But anyway, thanks for hanging out. With that, I'll sign off. Have a good evening.